Good morning, Himanshu. Good morning, Anu. I'm good. Good morning, Sophia. Your tour over? Still in tour. Sneha, good morning. Huh? Tour is not over. When returning? At railway station. Roll number one. How was the tour? Good morning, Ashwita. Good morning, Innovation. Good morning, everyone. Okay, better join the class, please. So now look here, we are going to start the session. We are going to write the most important parts and understand the most important sections. Now here what is exactly given? Last class we were discussing about heredity inherited. The traits which are inherited from the parents to offspring are called as inherited traits. So we have talked about somatic. Good morning Jivita and good morning Ashwita. So those traits which were inherited, that is exactly coming from the parents. As we know that okay, the traits which are inherited is coming from the parents. Next important thing what we have learned another important thing that okay, it is exactly the DNA which is carrying the genetic information from the parents to the offspring. Understood? So next important work what we have studied. Come on, one by one, start checking. After that, we have talked about heredity, that is the gene section we are going to discuss. Then second part we are going to check. So gene is function unit of heredity, present at a specific point on a chromosome. So if somebody will be asked you, this part is very important to understand. So let's check this one out. What in all things are present here and what in all. Yes, good morning. So you need to understand that the cells, chromosomes, and DNA all are intertwined. They're interconnected. So before reproduction, what happens? Before reproduction, the chromatin, that is the thread-like structure present, will be getting converted. The thread-like structure present in the nucleus will get converted. That is called as chromatin. Will get converted into chromosomes. The chromatin will get converted into chromosomes. The thread-like structure that is present in the nucleus will be exactly getting converted into chromosomes. I hope you might have understood. So now, after that next point, what we have to understand. So check this one out. So because biochemical gene is a small fragment of DNA. You remember biochemically, gene is what beta? Biochemically, what is gene? It is a small fragment of DNA. It is a small fragment of DNA, beta, and it is capable of replication. It is capable of replication means it is capable of making itself one, two, three, four more copies. Okay, so it is capable of making more copy of himself. So that's a major section. So the chemical basis of heredity is DNA. So what is the chemical basis of heredity? DNA, and that is called as what beta? Nucleic acid. So the chemical basis of heredity is considered as DNA. They will ask these questions. So specifically, all important points it is written. So here, good morning, Jotam. I Jotam online or offline? So definitely this is most important. Please check this one out. So we must remember this one. So biochemically, gene is a smaller fragment of DNA. Which is the smaller fragment of DNA? Gene is the smaller fragment of DNA. And it is what capable of? It is capable of replication, means making its own kind. Okay. expression and mutation. So what and all things it can do? Good morning Tarun, good morning Divya. So beta, what and all things it can do? It can express itself also and it can mute itself also. That is called as dominant and recessive traits. Okay. So chemical basis of heredity is what? DNA. So if somebody will be asking you what is the chemical basis of DNA, then the chemical basis of heredity is DNA, which is exactly consisting of gene. And gene is exactly capable of undergoing replication, mutation, and expression. And it is exactly carrying the characteristics. Okay. So the gene can be defined as a unit of DNA. Yes, it is a unit of DNA. Underline this one, note down this one. This is the most crucial part, please. So, on a chromosome, and which governs the synthesis of protein that control the specific characteristics. Yes, it is controlling what beta? Specific characteristics of an organism. The characteristics in an individual can be result of single gene or multiple gene. That's the most important thing. So whatever characteristics or trait a human organism is having, it can be due to one single gene or multiple gene. It can be due to one single gene or multiple gene. This is the most important thing. It can be due to one single gene or multiple gene. So we have talked about gene. So what are the 4 points I have discussed about gene? Can anybody explain it? Good morning, Beta. Beta, look here. You need to understand what is a simple part of mutation. Listen, Yashwin. Somebody will ask you what is mutation. Change in the DNA strand is called as mutation. Understood, beta? So, if somebody will ask you what is mutation, in the most simplest way, how you can answer what is mutation? See, the single strand is called as ribonucleic acid. Gopika, good morning, beta. Where were you from these classes? Are you attending the classes? So you need to attend the classes because marathon starting today, ninth revision starting from today. Sorry, 10th revision starting from today. 10th revision is there. First revision session we are going to start with chapters, science chapters, because science we are not having time in board examination. Okay, so today your first session of science revision is there. It will be late night also. You be patient and you just attend the classes. Micro test is going on. Okay, name is the nice. But yes, so here also today revision. Today very quick fast session will be there. So can I tell you the chapter name or can I make it secret? Anybody tell me. Should I explain you which chapter we are going to have first revision? Yes, 96 days left. So definitely we people are going to inform you that 96 days left. Every day we are informing this, huh? Which one? Which one? Which one? Come on, say. Life process. Easiest chapter you want to take. Why? Easiest chapter. Take some difficult part. Which chapter? Come on. Give me some that chapter which is exactly going to be problematic for you, which is troublesome chapter for you. Easiest chapter. Any chapter which is going to be tough. Life process is confusing. Okay. Other children, nobody is wishing for any class. Which class? Huh? Carbonates compound. Control and coordination. Okay. Control and coordination. So both goes for control and coordination. Two people are telling control and coordination. Two people are telling carbonates compound. Yes, carbonates compound. Na ethanol test and other tests are there. Na? We need to discuss. So teaching, asking question, we will discuss. Light chapter. Light chapter, we will have 1 week session. I'm going to give 1 week session for light chapter. There are kuti questions I'm going to discuss. Yes, light, magnetism, these chapters I'm going to teach in last part. Pongal is full dedicated to light and magnetism. Okay. So now look at it. Now Yashvit has asked me a question. First, let me explain Yashvit. So this is exactly called as DNA strand. Okay. So now in DNA strand, like this coding is exactly. So where in DNA strand, codings will be there. So better your DNA strand, the genes present will be there, and some like A, H, B, 1, like this codings will be there. Understood or not? So in this coding, a slight change in any one of alphabet is called as mutation. So that means the coding system will go corrupted. That is called as mutation. So usually what we are calling it as corrupted. Understood? Corrupted here means that means what we are saying. That means the system is getting corrupted means will not get the signal, na? But here it is not about signal. It is the entire generation. It is not the only signal. It is the entire generation. Understood? It is the entire generation. So you need to understand that this is not a single part. It's the entire generation system will be affected. Yashvi, did you understood what is the meaning of coding, beta? RNA is single strand. RNA is single strand, and RNA will be exactly single. That is called as ribonucleic acid. Got it? The double helix generated is called as DNA. Understood. The double helix is generated is called as DNA. RNA is single strand. So single strand will be there. In this single strand, ribosomes contents will be very important, playing important role. Class 9 have already taught this is exactly called as DNA. And a change in any one of the alphabets is called as mutation. So these are codings, beta. Change in coding is called as mutation. Understood? So change in a single coding will be coming as a mutation. I hope you might have understood. Everybody understood now. So that is called as. That's a very good question. Now look here. So gene can be defined as a unit of DNA. We have already understood what is the unit of DNA. Gene can be defined as the unit of DNA, and it is where present on chromosomes. So certain segments we must understand, and it is governing the synthesis of protein and control specific characters. Believe me, trust me. One very important thing is key metals and non-metals. Okay, we'll check with her. We'll check which one is there one by one. Priority I will do. Kavya, good morning. So now, sorry, Krish, Vishwa, good morning. So definitely, please pay attention here. So yes, half will be there, half characteristics will be there, but that is not enough to make the characteristics of an individual. It has to meet and make. Okay, mutation is a very specific characteristics. RNA, no DNA, it will be there, then it will be passed on. Okay, so gene can be defined as a unit of DNA, and with the chromosomes where it is present on chromosomes. So it will govern the synthesis of protein. So what I was, where I was is, कि whenever you talk about protein is a major section of every organism. You know, in coming future, what is going to be done means कि any organ, means all the organs, now they have a specific cells, understood, and these cells are generating what? Protein. Understood. The protein synthesis is one of the major work of every cell, specifically the nucleus. So what will happen in the coming future? Like if any human organ is there, so that organ is protein synthesis, protein generation of protein can tell the lifespan of that organ also. Are you understanding? If the so in future what will happen? We will exactly do one small work beta. We will take the protein of the cell, understood, of a specific cell of a specific organ, and will try to learn in what and all is exactly the lifespan left for that organ. Understood not? So the protein synthesis, the protein generation, the amount of protein generation, everything will be explaining about the lifespan or the health of the organ. I hope you might have understood this one. Now come on, we'll go for the next one. So this protein is a very specific one, and this protein controls specific characteristics. Understood? Or characteristics. What is the other name of characteristics, beta? The other name of characteristics is trait. Characteristics in an individual can be reserved as a single or multiple gene. So what character we are having, it can only be present because of one single gene also or multiple gene also. So that characteristics can be imbibed by a single gene or multiple gene. And there is the gene present, chromosomes. What is chromosomes made up of? DNA. Understood? So what is the unit of DNA? Gene. What is the unit of DNA? Gene. So one simple thing I want to teach you. Come on, if you people can see, if I can increase this one. Now, can you see better before now? Before we are telling that before reproduction, before the what you call that time, what will happen? That like contents will be present inside the nucleus, and better that is called as chromatin. Now, this chromatin will wind up like knitting we are doing now for making woolen things. Understood? So knitting is a process where we are making sweaters or everything. Understood? Same way knitting is done here. So how the knitting process is done here? This is exactly the process where the knitting is exactly done here. How? You see, Beta, these chromatin threads are there now. These chromatin threads are binding with histones. Histones are specific type of protein is exactly generated in the nucleus. Understood? So now these histones chromatins are present in the nucleus. Now these chromatins are going to wind in these 4 units of histone, up 4, down 4. So it is exactly going to wind together and knitting is done, and these knitting will be exactly making a structure called as chromosomes. And on the chromosomes, what is present, beta? Yes, chromatin. Actually, chromatin is the thread. Like this thread is called as chromatin. Understood? Not? So this chromatin thread will be exactly having DNA. Understood? Can you see the double helix? Yes, Shwet, can you see the double helix, children? Can you see the double helix, beta? This is double helix, what I was talking about. This is the base pair, got it? And in this Shamila, good evening, sorry, good morning, you missed a very specific portion. So this double helix will be DNA, and this DNA binding with the help of histone will make a structure called as chromosomes. And on this structure chromosomes, what is present, beta? Gene. What is on this structure, what is present is gene. So did you understood this part? So again I am repeating, inside the nucleus what is present, children? Yes, inside the nucleus what is present? Inside the nucleus the thing that is present is called as chromatin. Before reproduction, chromatin will exactly the thread like which is having DNA will wind up with histones. And beta, after that, what will happen? The histones will be exactly making histone will wind up and make one thread-like structure, and that is exactly called as what? Chromosome. And chromosome will have what, beta? Chromosomes will have very good, Yashwit, yeah, very good version. Super, beta. So that is exactly having gene. So gene is the unit of DNA. Gene is the unit of what, beta? DNA, which consists of specific characters. So this is exactly what we have learned. Got it? So now we are going to see. See, that's what I was talking about. Can you see everything? Can you see the coding system there? Children, can you see the coding system? So chromosomes is a thread-like structure. What is chromosomes? Thread-like structure present in the nucleus of a cell which carries gene. So during cell division, chromatin material condenses to form chromosomes. Understood? So here it is not chromosomes, it will be chromatin matter. So little bit change is needed. So this is exactly what called as chromatin. Chromatin is a thread-like structure. Chromatin is a thread-like structure. CHRO. Chromatin is a thread-like structure present in the nucleus of a cell which carries gene. So during cell division, chromatin material condenses to form chromosomes. Chromatin material is doing what, beta? It is condensing to form chromosomes. It is condensing to form chromosomes. So different organisms have different number of chromosomes. You remember, for human, how many is there, beta? 23 pairs are there. For human, how many are there? 23 pairs are there. There are thousands of genes on a chromosome. Remember this one. This is exactly thousands of genes are there. You know, beta, what? Thousands of genes are present. Thousands and thousands of genes are present which control various characteristics of an organism. And it is controlling what, beta? Various characteristics of an organism. So which controls various characteristics of an organism. In human being, 23 pairs of chromosomes are there. So total single if you will count, it is come 46. But pair means it will like pair of socks you are saying now, pair of shoes you are saying now. So that time it is coming pair means one pair is having two. So 23 pairs means total number of chromosomes is coming as 46. But beta, please remember one very important thing. Somatic cells, humans are having two type of cells. One cell is called as somatic cells, another cell is called as germ cells. Understood? Somatic cells and germ cells. So somatic cells are exactly having pair of chromosomes, but germ cells are having single chromosome. So chromatic, you remember this one. Somatic, in your cough, in your spit, in your cells, somatic is there. Okay, remember. It is what, beta? So, yes, Yes, that is exactly. So now we will be discussing what, can I repeat this part? Somatic cells will be there, beta, and germ cells will be there. So somatic cells are diploid. Remember, somatic cells are diploid. Diploid, Diploid. Somatic cells are diploid. Diploid means what? It is always present in pair. Somatic cells are diploid. Diploid means it is always present in pair. So where and all somatic cells will be there? Entire body somatic cells will be there. Only one place somatic cell will not be there. And that is what beta? That is the germ cells. Sperm and egg, ova. Sperm and egg are exactly, even the organs which are producing sperm and ova, they are also having somatic cells. Remember this one. So entire human body, entire organism bodies are exactly having what? Somatic cells. Okay, diploid means double. Understood? So entire human body are having diploid cells, but only one place it will be haploid, and that place will be what, beta? That single place is germ cells. That is called as, what is the germ cell name? Sperm from male and ova for female. Because the organs which are generating sperm and ova, like testis and what you called, testis is generating sperm, yes, and ovary. So these are also having somatic cells only. Understood? But these cells which are exactly going for reproduction, they are only haploid. Haploid means single. Haploid means single chromosome. So that means haploid and diploid. One time try to understand better what is the meaning of haploid and diploid. Once I write it, we will understand. So somatic cells, somatic cells and germ cells. Somatic cells, full body. This one only sperm and ova. So here chromosomes will be present in pair. These are chromosomes. So when chromosomes are present in pair, it is called as diploid. Understood? Full body except sperm and ova. Got it? So now diploid. Diploid means pair. Now here it is germ cells, sperm and ova only present, and it is exactly single. Got it? And it is exactly called as haploid. It is called as what, beta? Haploid. So please note down, one will be in pair, one will be single. So one will be diploid, one will be haploid. I hope everybody understood. Is the class okay? Everybody understanding? Full body will be there except sperm and ova. So this is a very important thing. Single terms are there, beta. You must all understand that. Then only in exam you will write exceptional. You will write better than others. Understood? So I hope everybody understood this one. Somatic is haploid and germ cells are diploid. Monkey data. So somatic is haploid and germ cells are diploid. So you must remember this one. This is the most important section, sir. If you are coming 24 minutes late, means it is not good. So you are missing 24 minutes classes. I hope now is it clear? Now is it clear, beta? So entire body inside the nucleus, you will be saying. So inside the nucleus, you will be saying double like this. How many will be there? 23 pairs. How many will be there? 23 pairs. Now because this 23 pairs, now it is also deciding. I will explain how it is exactly deciding everything. So this is 23 pairs will be there, and yes, 23 pairs will be there, and this is present in the full body except where, beta? Except sperm and ova. Yes, understood that, beta? Very good, beta. So now we are going to understand the next part. So now we are going to see this. So this is cell inside cell nucleus, nucleus inside chromosomes, and chromosomes is made with gene. Can you see the gene pattern, beta? Can you see the binding? Can you see the thread? You see the DNA is exactly having the unit of DNA is considered as gene. And here beta, can you see the gene? The single strands are there. Yashwek, see here. So the single strands are there. Is there coatings are there? Can you see GA how they have written? Got it, na beta? Together accumulatively they are called as DNA. DNAs are binding with histones. DNAs are binding with histones to make chromosomes. Understood? So in this thread-like structure, it is what is present? Gene. Is it crystal clear, children? The pictures are explaining everything. Correct, na? So is it crystal clear? Inside cell, what is there? Inside cell nucleus is there. Inside nucleus, you will get chromosomes. But beta, how chromosomes will be there? Understood? How chromosomes will be there, beta? In that chromosome part, you will be seeing gene. Gene will be exactly present on DNA. Understood? So this is the most important section. Now we are going to talk about the next section. Are we ready for the next section, children? Now you check this one out. Now look here, everyone. Why are these strands in different color in this pic? Because strands are of different color because what you call, that's why we discussed now, the strands will not be of same color, and the different color is indicating different type of coding. Different colors are indicating different type of codings, and also the gene, because there are thousands of gene present there. Which one will be selected, we also don't know. Understood or not? So thousands of genes are there. Which one will be selected, that also is not known. I hope everybody might have understood. So heredity, center of heredity. So okay, center of heredity in a cell. So what and all things are there? Now let's talk about in prokaryotes such as bacteria. The genetic material exists in an enclosed, unenclosed but recognizable area of cell, that is called as nucleoid region. Please remember this one. This is the nucleoid region, beta, where you can see that this is a very well settled cell, but that is not a very good cell because it is prokaryote. All these are prokaryote. In that, the DNA is not double coated. Understood? So it will be exactly a very premature content. It will be nucleoid cells. That part will be present where in nucleoid, and only one single DNA will be there. One single DNA will be there, but here multiple parts are there in nucleus of multicellular organisms. Understood? So here in eukaryotic organism, the genetic material exists in membrane bound, that is double coated. That's what I said. The in eukaryotic it will be exactly double coated. In prokaryote it will be single coated. Understood now? So it is not coated also. It will be exactly unenclosed. It will not be sometimes enclosed also. It will not be enclosed also. I hope everybody understood. Come on, say. In virus, which are not true cells, the genetic material is, that is in-seated in protein code. Where it is in-seated, beta? In protein code. Remember this one. This is the most important thing. Why virus? Ajit, tell me. Can you tell me one thing? What is speciality of virus? Why virus is different from others? Can anybody tell me? Who can answer me this question? How can we answer virus is different from bacteria? Can you tell the difference between bacteria and virus? Virus depends upon host. Other organisms. Virus depends upon other organisms. Okay, one answer Ovian has given. Who can give me better answer than this? It is not a true cell. Yes, version. It is not a true cell. But why? So only I'm asking now, bacteria and virus, what is the major difference between them? What is the major difference between a bacteria and virus? Who can answer? Now, it is not, it's outside, it is not live outside. That's the most important thing. Very good. So, three of you children, please pay attention. So, that's the correct answer, children. Please pay attention for this. If somebody will be asking you about virus, you will say that virus is dead outside. Outside the human body, virus is dead. Understood? So virus can be differentiated by their structure. Example, enveloped by non-enveloped, okay, genetic material, okay, the type of host they infect and their mode of. But the most important thing is still unanswered here, that virus is dead outside. The moment it will go inside the host body, that particular moment, what will happen? It will show the sign of life. But how it is? See, it is not helping its nourishment or something. It is just responsible for duplicating and replicating. So whatever life the virus is getting from the cell, yes, absolutely correct, Imanshu. So whatever life the virus is taking from the cell, it is exactly using it for reproduction. Understood or not? It does not show the sign of life. But the most important thing is it is duplicating itself and it is having its own protein synthesis. It is having its own boundary. Understood or not? So virus boundary is very difficult to break, because if any antibacterial medicine you will take, definitely you will have recovery. But antivirus, when you are taking, means antivirus should affect a specific virus. Understood? So it is not like you are taking in general. See, antibacterial medicine, if you are taking, means good bacteria, bad bacteria, all in your body will die. Understood? Not because you are taking antibacteria, more or less the symptoms, the contents will be same. But what about the virus? When you have to take virus, means a specific type of antivirus you have to take. Are you understanding? Which can cure. So for COVID, we cannot take AIDS medicine. Correct now? Both are generated from virus. Cancer, for cancer, we cannot take COVID medicine. Are you understanding? I hope people can be able to relate with this example, that we can't be able to take the medicines of virus. Virus are exactly enclosures of a specific type of organism. And you know what? Yes, and they will not be behaving the same way. They will be behaving in a different way. I hope children you might have understood. So now we will go for the next one. Today Vishwa is not there. Antivirus is dead or weaker virus. Absolutely correct. So antivirus is a dead or a weaker virus. Correct, Himanshu Beta. So here antivirus will be what type virus? Dead or weaker virus, and that's what it is injected in the body. So polio will be there and different type of what you call measles, the virus infections. Now for that what we are giving a type of antivirus medicines. Like we seeing a small children, what they are getting, they're getting injections. Understood or not? So these injections are exactly where the virus or the what you call that foreign agent is embedded inside the body. So by seeing those virus, our body will generate a true virus, a true antibodies. Understood or not? But that virus which is coming inside the body, it will be dead or paralyzed. It will not do anything. But let us see, in your house, a thief entered. Now we don't know the thief is exactly paralyzed thief or dead thief. But what will happen? We will take our weapons to fight with that. Correct now? We'll take our weapons to fight with that, thinking that the thief is a real thief. That's a true answer, beta. Very good, Rashmi. Everybody, please understand this one, and thank you for liking the class and thank you for understanding and saying at least yes. Okay. So now here in eukaryotic organisms, let's talk about the eukaryotic organism, the genetic material. Yes, Vishwa. Very good, Vita. But another one Vishwa is missing, Nare. Thank you for liking the class, Vita. The genetic material exists in membrane bound. So the nucleus will be double coated. Nucleus will be what? Double coated. That is eukaryotic, Vita. Nucleus will be double coated, then it is what it is called as what you call eukaryotic organisms. Nucleus will have a double coating. Can you see nuclear envelope? Last year I have taught all this with you, remember, Peter? So in virus, which are not true cells, the genetic material is what you call seated inside the protein core, constituting the viral head or capsid. The genetic material in case of virus can be DNA or RNA. That's the most important thing. Yes, the genetic material. The genetic material will be exactly in case of virus can be DNA or RNA. That's the most important part here, beta. Vital, good morning. Nowadays I'm not seeing your name, beta. Where are you? So now we will go for the next one. This I think this is the last. We are going to go for the next seat. So this is still here. We have exactly understood the part, huh? So that's about the virus. It is not a true cell. Already we people know that it is not a true cell. The genetic material is what we call seated in the protein code. That's what. And with they believer of what? They are believer of only one thing. They are believer of duplicating and replicating once they are entering inside the host body. Yes, absolutely. So we'll go for the next slide. So now we will specifically discuss with about gene and specific definitions. Okay. So beta, what is the meaning of alleles? What is the meaning of gene? Now we are going to understand. Okay. Now Mendel has proposed inheritance. But inheritance means what? When we carry the genetic information from our parents. That is inheritance of characters. That is inheritance of characters. And no single individual. See, at the time of understanding of cells, we learned a very important part, and that is every cell exists from a pre-existing cell. Understood? So whatever is generated, thank you for liking the class beta, whatever is generated, that is generated because of the pre-existing cell. Understood? So accidentally one cell came in what you call earth, and definitely it will keep on making new cells. What a genome type is also I'm going to explain beta. Gene coding. Gene coding will be specific gene codings are called as genome beta. So we are going to discuss that also. So now let's one by one we are going to understand. So now yes. Now it is gene. So when genes will be exactly, so Mendel proposed that inheritance of characters is controlled by paired genetic unit or factor, and it is exactly called as what beta? Gene. So what is the gene? Gene will be exactly paired genetic units. Paired genetic units. Understood? So this pairing is a type of comparison. You need to understand the comparison between the father and the mother part. Understood or not? The father and the mother specific characteristics will be exactly compared. Understood? So now we are going to talk about that also. So this is now called as gene. So please remember what is gene. Yes, what is gene beta? It is proposed that inheritance of character is controlled by paired genetic units. That is paired genetic units will be exactly called as what beta? Genes. Now we will go for the next part. They are present in all body cells. Where it will be present beta? It will be present in all body cells and are transferred from one generation to next generation. So if we will be talking about a species continuation of in a planet, then definitely the species should have gene to carry the genetic information from one generation to another generation. Understood? So now gene work in pair and are represented in letters. That's what I have discussed in the beginning part. When Yashvik has asked me, Auntie, where is the gene present? I have discussed about the gene present in the double helix of the DNA. Understood? And in that gene, did you saw that A, B, G, H was written? And that was exactly in that part. We have seen that it will be present with letter alphabets. Okay. The letter T and T actually represent two forms of genetic control of height of an organism, and there only beta things will change. Absolutely correct beta. So two forms of same gene that control the characteristics of organisms are called as alleles. Alleles and alleles. These are exactly important. So dominant and recessive traits will be exactly always in pair beta. Always in pair, it is called as alleles. Understood? So remember this part. We have started from cell. In cell, we learned chromatin. Chromatins will be exactly binded. In chromatin, what we have seen? We have seen DNA. DNA single unit is called as gene. Now this will be binded together. It will be chromosomes. Now inside the chromosomes, what and all are there? So we understood that gene will be exactly always present in pair. Good morning, Kavya. Gene will always be present in what beta? Pair. So gene will always be present in pair. So it is paired genetic units or factors, and it is exactly called as genes. Understood? So now next part beta, they are present in all body cells, and it will be transferred from one generation to another generation. Yes, beta. Can you repeat last point? Yes, I am going to explain the last point beta. Yes, Yashvik. In RNA also you might be seeing beta, but it will be half only. It will be half only beta. Ribonucleic acid will be there na. Yashvik, it will be half. I'm going to I'm going to explain that part. Please check this one out. So they are present in all body cells and are transferred from next generation during reproduction. Now, beta, next one. Gene work in pair are represented by letters. That's what beta. In RNA also you will be seeing yes with half part will be there. There also beta RNA part it will be having. Okay. The letter T and T are actually representing two form of same gene. But beta one capital letter is always representing which type of trait? Dominating trait. And the small letter is always representing recessive trait. That's the difference. The capital letter will be representing what? Dominant trait, and the small letter will be representing recessive trait. The two forms of same gene that control the same characteristics of organisms are called as alleles. And because in the beginning itself I told na eye color, when somebody will be asking you your eye color, so parent gene and both the parent father and mother eye color will be exactly giving, correct na? The selection will be exactly responsible, but it should come in pair. So one should be coming from father side, one should be coming for mother side, and that's what Navita one question was asked. So here by mistake to typing error will be there. So that was one question has been asked. You just distracted me, girl. So I was about to explain this part, beta. This part is what I was discussing. Yeah, in alleles, you need to understand that whenever we talk about alleles, we need to understand it's a specific characteristics, and it will be exactly coming from father side and mother side. One question, yes, one question was asked, like parents are exactly the sperm and the ova are coming and they're fusing and making zygote. So definitely the child born will have the characteristics of both father and mother. But is it possible that total 100% characteristics can be taken from father or total 100% characteristics taken from mother? Is it possible? Say yes or no. Thank you for liking the class because say yes or no. Absolutely correct. Very good, Yashwin. You didn't wasted time. So Kavya also didn't wasted time, and then Anu and Ravashan. So yes, the correct answer here is beta. The correct answer is yes, Jyotam. It is no. Why beta? Because the genetic variations are exactly getting selected according to dominant and recessive trait. So alleles are paired. Alleles are paired characteristics, and from there, which one will be collected, we itself don't know. I hope you might have understood, beta. So here it will always be in pair. So now, beta, two forms of same gene. Here we will cut this part. I will write two forms of same gene. Wait. Same place to writing error. I need to discuss with my team. So two forms of same gene that control the same characteristics of an organism are called as alleles. So alleles can be of how many types? Dominant and recessive. Now is it crystal clear? Dominant. Dominant and recessive. So what are the two traits beta? Dominant and recessive. Now is it very clear? Is it very clear? Is it very clear, beta? That alleles, every time they are telling what is alleles, the pair of characteristics are considered as alleles. And alleles are of how many types, beta? Alleles are of two types. What are the two different type of alleles, beta? One is dominant and another one is recessive. Understood? So we have come to a conclusion. So from the beginning, whatever map we have generated, let's check this one out. Let's check this one out because we should have some conclusions now. So conclusion section, how we are going to generate inside the cell nucleus is there. Understood? In this nucleus, thread-like structures are there. These thread-like structures are called as chromatin. Now, because this thread-like structure, if you will see from microscope, it will be DNA strand. Correct? Huh? Now it will not be single, it will be double. Ribosomes will be, sorry, RNA will be exactly double. Understood? So here dominant traits beta. Okay. Downtown lag आ रहा है बेटा. एक बार नेटवर्क चेक कर लीजिए आप. नेटवर्क चेक कर लो बेटा. So chromatin is there. Understood? So when you are talking about chromatin beta, this chromatins are thread-like structure. And these chromatins are exactly DNA, correct now? Now this DNA will bind with histones. 8 pairs from front and back, 8 pairs of histones they will wind up. Understood? Now in this DNA, the unit of DNA is called as genes. Understood now? So now this will wind up and it will make chromosome. Understood? So this is called as telomere. Understood now? Telomere means the corner part. Understood? The center region and corner is having different names. Okay? So now beta, this is chromosome. In this chromosome, if you will see, so another chromosome will be there. So this is one characteristics and this is another characteristics. Understood? So these are genes. These are genes. Now these two genes, one will be either T and one will be T. The selection will be done accordingly. Understood? The selection will be done accordingly. So usually what happens now beta? कुछ नहीं होगा शेर की तरह आंसर लिख के आओगे कभी डरो मत बेटा. So now look here, this is exactly coming like this. So the what you call the thread will be exactly winding like this. Got it? So genetic threads will be exactly winding and characteristics are selected. So let's see. So I hope everybody now understanding DNA is consisting of what beta? Gene. And pair of genes will be considered as alleles. Alleles are of how many types? Two types. One is dominant and one is recessive. Is it crystal clear? Gene pair is called as alleles. Alleles are of how many types? Two types. One is considered as what beta? One is considered as recessive. Another one is called as dominant. So I hope till here everything is clear. Yes, alleles are of two types, beta. What are the two type of alleles? One is dominant, another one is recessive. Now, alleles of tallness is represented by capital T and small t. I hope everybody understood. I'm from Chennai, beta. So it is capital T and small t. Now, beta, we need to understand which allele can exist single and show characteristics. Yes, that's what beta. That's what whenever at the time chromosomes from the chromosomes, whenever we are talking about, it will be selected there. Vishwa, Yashwat. So now look at beta. One time you understand this one. So DNA, if this single P is there, this is very very powerful. Understood? Dominant. So only its name is got as dominant trait, and this one is called as recessive. So whenever dominant and recessive will be there, always dominant is selected. But when two recessive will be there, then only recessive is selected. Then only T is selected. Otherwise, in all the cases, which one is selected, beta? In all the case, dominant will be selected and recessive will be selected only in one case if both are recessive. If both are recessive. So this is one of the most important things. So now better look here. Now in our country or in the world, there are certain traits which are very very dominant, and most of the human population. How we did this survey? We did this survey by exactly checking what is the major population of humans are having what type of hairs. Understood not? What type of skin color? What type of earlobes? What type of eyes and tongue rolling? Many people they can roll your roll tongue, and many people cannot roll tongue. Understood? So that is also dominant. So here curly hair is dominant. So most of the people are having, I don't know beta, mostly February I may come. Most of the people are having what type of trait? Curly. Understood? So here can be curly. So it is dominant. Understood? So you might be seeing most of the boys they are having curly hairs. Very ratio is 3:1. Correct only beta. But here the most important thing when we talk about curly hairs are exactly the dominating one. Next is the skin color, normal skin color. Albino is exactly recessive. Correct? Now, earlobes, free earlobes. Many people of the world is having what type of earlobe? Free earlobes. Eyes, brown and black color is major dominating one. Tongue rolling, ability to roll tongue, inability to roll. It is also which one is having massive beta? The one which can be able to roll the tongue. I don't know many people. I don't know how to do it, but definitely many people can roll the tongue. Many people cannot be able to roll the tongue. But major part of the population can roll the tongue beta, and this will be considered as a dominant trait. Okay. Now, homologous chromosomes. This is a very important thing. Those who are preparing for NEET and those, yes, beta, dominant traits. So now those who are preparing for NEET and those who really wish to understand, are you seeing that identical chromosomes? Can you see the identical chromosomes beta? This center part is called as centromere. When one chromosome got connected to the another chromosome, the part is exactly called as centromere. And this portion when they are free is called as telomere. What it is called as telomere. Center part is called as centromere. The free ends are called as telomere. Center part is called as centromere. The free ends are called as telomere. Absolutely super beta. So now look at homologous chromosomes. Now we are going to understand about homologous chromosomes. So these are chromosomes pair of approximating same length. Understood? Now centromere position and staining pattern with genes for the same characteristics at the corresponding locale. Means this is identical. Why? Because centromere location is also fixed and the telomere free ends are also same. So they are exactly considered as what? Homologous organisms. So in an organism, one homologous chromosome is inherited from mother and the other one will be from father. This is the most important thing, beta. We must always remember. One is collected from father, another one is collected from mother. It is always be in pair, but one will be coming from father and another one will be coming from mother. So it is exactly taken from father and mother. This is most important thing. I made it very simple for all of you, isn't it? So, beta, are you all understanding? Anybody, anytime when you will see these videos, you'll be able to understand the content very easily. Correct, na? We didn't make it tough. We make the chapter very, very easy. Yes, na? Yes, you people have contributed and asked very good questions. Then only this video became good because most of the questions, important questions, you people have asked. What is DNA? How it is looking like? What is mutation? And what and all things are present there? What is gene? So definitely, this part will be helping other children also. Yes. So now we are going to go talk about the next slide, beta. So now we are going to go for genome. One child asked me genome. Okay, so now I'm going to talk about genome. That's what I told the beta. I'm going to explain about genome. Just give me some time. So now, beta, what is the meaning of genome? The total number of genes present in haploid set of chromosomes are called genome. Understood? Or gamete will be exactly considered. Haploid means beta single chromosome. Single chromosome. So the total number of genes present in the haploid set of chromosomes. Haploid means single set of chromosomes. So in single set of chromosomes, why beta? Because they are most crucial and they are only going to participate in reproduction. Understood? So they are only going to participate in reproduction. So they are exactly the most crucial one. I hope you might have understood this one. So what is the meaning of genome, beta? The total number of genes present in the haploid set of chromosomes or gametes are exactly called as, ma'am, in homologous chromosomes, there in our syllabus, no, beta. Just for letting you understand. You might have seen, how many of you have seen Harry Potter? In that they are saying the pure blood and mixed blood. That is the purity. So when all the alleles will be exactly having same type of trait, it will be considered as the pure breed. Understood? So for breeding, I'm just going to I will just explain that part. Given it will be same type, it will be considered as the pure one. Understood? Yes, pure breed. Yes, the total number of genes is exactly present. All the best, we will write nicely. No problem. So now what is the total number of genes present in haploid set of chromosomes is exactly called as what, beta? In the gametes or gametes is exactly called as genome. So it is not present anywhere, it is present in the gamete. Gametes are of two types, one male gamete and female gamete. Male gamete is called as sperm and female gamete is called as ovum. So how we are going to just see one time so that confusion will not be there. So that means this is a haploid chromosome. Only one single strand is there. Now, how many total number of genes are present in this? How many total number of genes are present? Count the total number of genes here. The total number of genes present on a the total number of genes present in a haploid chromosome. Haploid set of chromosomes. The total number of genes present on the haploid set of chromosomes are called as genome. Understood? Now, beta, what is the meaning of progeny? Now, we are going to see some very important name. Okay. Yes, beta. Homologous means beta identical. Yes, beta. Homologous means identical. So when somebody will be asking you what is homologous means, beta, two dominant traits will be there. All the traits will be very strong enough to express in the progeny that will be considered as dominant homologous. When the chromosomes looks particularly identical, means the father and the mother, they will be exactly joining together, the chromosomes joining, it will be fully identical from the center part and to the common telomere region. So it will be exactly identical. That time it is called as homologous, beta, pure breed. and homologous are always associated with pure breed. I hope it is clear, beta. Now, offsprings means the babies are exactly called as progeny. Understood? Now, factors. These are the gene located on the chromosomes. This is very important. So, factors. These are the genes located on the chromosomes. Now, locus. Locus means what? The location of a particular gene in a chromosome is called as locus. Understood? Next is allele. It is exactly two alternate forms of gene. Understood? So one will be recessive, another one will be dominant. And that's what the pair of genes will be present in that what you call chromosomes, that will be considered as what? Alleles. So homozygous or homozygote. This is very important. Now, Yashwit, your question I am just explaining. So, what is the meaning of homozygous or homozygote? Now, look here, beta. Homozygous and homozygote and homologous. Everything we are going to discuss. Now, look here, beta. So, an individual with identical alleles at one or more loci on the homologous chromosomes is called as homozygous. So beta here, if you will be seeing the chromosome strand, you will be seeing same identical characteristics will be there. Same identical characteristics will be there. It will be considered as what beta? So same homologous chromosomes will be present. Identical characteristics will be there. Then it is called as homozygous or homozygote. Note down. It is considered as homozygous or homozygote. Homozygous or homozygote. All the best. All the best. So this is called as homozygous. So now better understand this one, everyone. Loki means beta a fixed point from where whatever distance you are taking, it is exactly same. That is Loki. But here, this is not in that. That is in mathematics. Here, locus means the location of particular gene. Understood? And beta, here it is not only one location. There may be the particular gene can be present in more than one location. So if the particular gene is located more than one location, it is called as the plural part is called as loci. Understood? Locus is a single and loci is plural. I hope you might have understood this one, Beta. So locus is single and loci is plural. Plural is loci is when more than one is present. It is considered as loci. But in single, it is considered as locus. Till here, is it clear, beta? Single, it is considered as locus, and when it in pair, it is present, it is considered as what? Loci. I hope everybody understood this one, beta. Now, homozygous and homozygote understood. So now, one time I'm going to show you one thing. Identical alleles. Can you see? You see the strands, the DNA part is totally identical. Can you see the color strands? The color strands match with each other now. So only it is called as homologous. Understood? And so only the homozygous means when both are exactly having all the identical features. That is called as homozygous. Children, is it clear now? यशवित ना इज इट क्लियर बेटा देखो यहाँ पे दोनों का कलर देखो हर जगह कलर और स्ट्रैंड का लोकेशन सेम है everywhere color and strand locations are same beta. So what we are going to conclude it as? We are going to conclude it as homologous or homozygous or homozygote. Super. So homologous, homozygous will be coming homozygote and homozygous. Yes, it will be coming from homologous pair only. Thank you for liking the class. Thank you for attending the class, Peter. It was indeed a very important class. It was very, very, very important for you children to understand this part so that nobody can confuse you. Nobody can say this is there, that is there. But you are enough to teach them. So I think you people are enough to teach many people. Yes. So definitely if somebody will be asking you about homozygous, homologous. You'll be able to answer better. And here beta homologous is exactly homozygous and homozygote. That's the most crucial part. Now we'll go for the next one, beta heterozygous. Heterozygous part will be there. So when heterozygous somebody will be asking about, then you remember an individual with different alleles. That time the strand color will not be same. The strand color, strand height will not be same. That means what? Can I draw and show you the? So now look here, so that you will be able to understand. So that time it will not be same. One chromosome will be like this, one chromosome will be like this. So definitely it will not be same. So that is considered as what beta? Homozygous or homozygote. An individual with different alleles. An individual with different alleles or on one or more loci is called as homologous chromosomes on the homologous chromosomes. Understood? So pattern will be different on homologous chromosomes. Then it will be exactly now coming under as heterozygous and heterozygote. Heterozygous and heterozygote. So when the alleles will be like this, beta, T and T. So this is not same, this is different. This T and T are different. This T and T is not same, but these are homozygote. Correct now? So now we can quickly recap. We can do T and T. It is homologous. It is what? Homologous. I am a key. Stop using mobile. Oh, it is homologous. Understood? It is homologous and it is become homozygous also. Homozygous or homozygote. Beta because same traits is there. Small t, capital T and capital T. But when mixed will be there like this, it is called as heterozygous. Mixed will be there, it will be considered as what beta? Heterozygous. I hope you might have understood. The moment we are doing mix of it, it will be considered as what beta? Heterozygous. I hope everybody understood. Yes, so that was very important. Very important. Yashwit Samash, may I beta? Everybody understood now. So dominant alleles, the which one will be expressed is called as dominant. Which one can be not expressed? Only pair will be able to express it. It is called as recessive. Understood? Recessive. No doubt, no doubt. So genotype means the specific allele in genetic constant is called as genotype. Phenotype is physical appearance. Genotype is genetic and phenotype is always remember that. So genotype, the specific allele or genetic constitution makeup of an organism is called as genotype. Phenotype is the observable characteristics or physical appearance of an organism is considered as phenotype. Did you understand what is the specific difference between genotype and phenotype? So genotype, gene, and phenotype is physical. Got it? So what you can see is phenotype. What is present inside the body constituent is called as genotype. The genetic constants will be exactly considered as genotype. You see, the specific allelic or genetic constitution makeup of an organism is called as genotype. It is makeup of an organism is considered as what beta? Genotype, and phenotype is observable characteristics. Phenotype is what? Observable characteristics. Phenotype is observable characteristics and genotype is exactly present inside constants. Phenotype. Kidding? Yeah, father of genetics. Mendel is supposed to be the father of genetics. Now I'm going to wrap it up here till here, beta. Thank you so much for joining the session. Thank you so much for liking the class. Thank you so much for everything. One more time, we will be checking from the beginning. 5 minutes, we will do revision. Okay. Thank you, children. Today also evening class will be there. Maths now. Yes. Thank you so much for joining this session, children.